Hey guys, Chapter 5 vocab video here. Chapter 5 is all about viruses. So viruses have very few parts. The outer part of the virus, for most viruses, is the capsid. The capsid is made of proteins and it can take on different shapes. A lot of times it's icosahedral, which is like a 20-sided dice. Sometimes it's spiral shaped, but it's always made of repeating units called capsomeres that make up the protein shell. I sort of think of the capsid as like the candy coating of the virus. All right, sticking off of that candy shell or protein shell of the capsid, most viruses have other proteins that sort of decorate their surface, that stick off the surface of the capsid, so we call them protein spikes. And these different spikes identify different viruses. For example, with flu, two of the spike proteins it has are hemagglutinin, or H, and neuraminidase, or N, which is why you hear a lot about H1N1 or H5N9. The different strains of flu virus have to do with what spike versions of spikes they have sticking off their capsid. Enveloped viruses actually contain the capsid inside a membrane. The envelope is a membrane. It's a cell membrane, a phospholipid bilayer, and it's actually taken from the cell as the virus leaves the cell. But it is decorated with spike proteins of that virus. So the virus-infected cell, the virus will direct proteins to the cell surface, and then when it leaves, it'll take a bit of that membrane with it, already been decorated like the virus itself. Viruses that only have a capsid and no envelope are called naked viruses versus those with an envelope, which are enveloped viruses. Some viruses contain their own unique enzymes, one of which that's pretty important is reverse transcriptase. So retroviruses like HIV contain this special enzyme, and what it does is it allows them to take their genome, which is RNA, and it converts it back to a DNA. So in our cells, we only have the ability to make RNA from DNA. We can't make DNA from RNA. The only enzyme that does this is called reverse transcriptase, and it comes from viruses, but this enzyme has actually been used a lot in biotechnology, so it's been really useful for us in a technology way. When viruses infect cells, the first thing they have to do is adsorb to the cell. So it's not a B, it's not absorption, it's adsorption, which means to attach or stick to the surface of something. And that's the first step. Viruses are usually very specific for the types of cells that they like to infect, and they, roam around until they find the right cell, the right surface to adsorb to. Then they enter the cell. That stage is known as penetration, and there's a couple of different ways that a virus can penetrate the cell. Once it is inside the cell, it will uncoat. In other words, the envelope will fall apart and the capsid will open up and release the genetic material of the virus into the cell. Now, viruses are too small to see using a light microscope, but we can see cells, infected cells with a light microscope, and we can tell if they're infected because viruses make the cells sick. And those symptoms that the cells have, I guess we call cytopathic effects. It literally is a word that means disease of the cell, pertaining to disease of the cell. So one of the things that happens when cells are infected is they die. And so if you have a cell culture and there's cells all over the plate, in a lawn all over the plate, and then when you infect it with virus, you look again a day later and now there's like holes and gaps and regions where the lawn is dead. All right, that's a cytopathic effect. We call those dead zones plaques. It's another word that I have for you right here. Right? Plaques are dead zones of cells. We can see these with human cells and even bacterial cells when they're infected with viruses. All right? Another cytopathic effect, or a sign of viral infection, disease of the cell, are syncytia. Some viruses make cells very sticky, and they start to stick together, and then they fuse together. So when you look under the microscope at that lawn of cells, now you have patches where the cells have actually clumped together, and you have like giant cells with multiple nuclei. Those are called syncytia. Literally means, syn means together. Another way that viruses can cause disease when they get into cells is they can insert themselves in the DNA or control, otherwise find ways to disrupt the cell cycle and cause cancer. Basically, cancer is just a cell reproducing uncontrollably. So oncoviruses, onco means cancer, oncoviruses are viruses that can cause cancer, that can make a cell, transform a cell into a cancer cell. So HPV is an example of an oncovirus. It can cause cervical cancer, anal cancer, oral cancer, several different types of cancer actually, and it is vaccine preventable. Other common ones are some of the hepatitis viruses, Hep C and Hep B. There is a vaccine for Hep B, not yet for Hep C. Bacteriophage. This is a virus that specifically infects bacteria. It's a little confusing because it's called a bacteriophage. Phage is something, it means to eat or swallow. And it sounds kind of like macrophage, which is a white blood cell of the immune system which eats bacteria to protect us. Bacteriophages may be a little bit of a misnomer, but it's what we call them. So when we talk about phages in microbiology, we're actually talking about these little viruses that specifically infect bacteria, only bacterial cells, not human cells. And you're going to watch an interesting TED Talk at one point about how bacteriophages could be the future of antibiotics, using viruses to infect and kill bacteria rather than drugs. Temperate phages are ones that can enter a bacterial cell, little bacterial viruses that enter the cell, and then they can insert their DNA into the bacterial cell and sort of stay there quietly, dormant, not producing more viral particles until they're triggered to do so. So if conditions are not good, they might think, we will ramp this infection up at a later date when there's more cells around for us to infect. So temperate phages are sort of in a dormant, they have this ability to go dormant. And when they're in that dormant state, that state is called the lysogenic state. So lysogeny is dormancy, whereas the lytic state, or lysis, is the state where they're actively producing more phage particles and erupting from the cell. So this lysogenic state is non-fatal to the cell and can last for a very long time and is also vertically transmissible. So if a bacteria has a virus in a lysogenic state and it duplicates itself, it's actually duplicating the virus as well, quietly. So then, when something activates and triggers the virus to go into the lytic state, suddenly all these viruses start bursting out of all these bacteria that may or may not have known that they were infected. Human viruses can do this too. We'll talk about that more in class. It's called dormancy or latency rather than lysogeny. Lysogenic conversion is that process of going from the lysogenic state, that dormant hiding state, to the active lytic state where they're actively producing virus particles or phage particles. And then lastly in this chapter we talk about prions. Prions are sort of their own class. They're not viruses, they're not bacteria, they're like less than viruses on the scale of what is a living thing. They are literally just proteins. They're proteins that are malformed, and then they can go around and cause other proteins to malform. And then these malformed proteins clump together and end up killing the cell. So they are sometimes called infectious proteins because they can be transmitted from person to person. There's only a handful of prion diseases that we know about right now. Creutzfeldt-Jakob disease, which is the human form of mad cow disease, is one. They all affect nervous tissue, so they're all in the brain and they cause nervous system issues. But they are infectious proteins. I like to do an analogy where I say they're kind of like zombie proteins. That's all.